So in this video, I'm going to quickly teach you guys about what a karyotype is, and I will show you kind of how a karyotype is made. So to start off, a karyotype is basically a picture of all of your homologous chromosomes paired up. So they use the size of the chromosome, the way that the banding pattern looks. So if you can see this picture, all of them have different kind of colors. They've used a dye to kind of color the chromosomes. The banding pattern would be all of the different bands on them, and the centromere position would be kind of the middle point on that specific chromosome. This is a picture of chromosomes that are not replicated. They're individual chromosomes, and they're matching up our homologous pairs. So this would be chromosome pair 1, 2, 3, 4, 5, etc. And then the last pair is your 23rd pair. So the reason that we would use a karyotype is because we would want to maybe during a pregnancy, we want to figure out if there's any genetic abnormalities. So maybe an extra chromosome or one less, or maybe chromosomes that are missing pieces of it. And we usually do this during about the 3rd month of a pregnancy. So when we are making a karyotype, we take a blood sample. So we either do amniocentesis or chorionic villus sampling, which we talked about in chapter 14 and 15. Those are both ways that we can get a sample of DNA from a baby, or blood. We can take a sample of blood and we centrifuge it, which means that we put it in a machine that spins it really quickly. All of the really heavy stuff goes to the bottom and all the light stuff stays at the top, so that they can easily separate the blood cells out, and then they can take DNA from the cells. So the white blood cells are then basically treated so that cell division stops. We're not looking at different stages of mitosis, and we want to make sure that we stain them, take pictures of them underneath a microscope, and then eventually we use a computer program to arrange them into the pairs. So for Bio30, instead of using a computer program to rearrange them into their pairs, we're going to be doing that ourselves. So you guys, after you're done watching this, will do an activity using a computer kind of animation. Normally we do this in class where we cut and paste things on our desks, but obviously we're not doing that this semester. So just a little bit more about how we obtain it. Remember that it's either chorionic villus sampling or CVS, or we can do amniocentesis. Those are 2 ways to actually obtain the cells for a karyotype. And then lastly, just a reminder about our homologous chromosomes. We have 23 pairs of chromosomes. So again, this is showing a cartoon version of our chromosomes. This time it's showing unpaired chromosomes. And just a reminder that the 23rd pair would be the sex chromosomes. So if it is a female karyotype, they would have 2 X's. They're not actually X chromosomes. Okay, they only look like X's if they're replicated. And then if it's an XY, it's actually just an X, which is a large chromosome, and then the Y is just a lot smaller than the X chromosome. So it's going to be kind of like, this is technically a non-homologous pair because they're not actually carrying the same genetic information, in that they're carrying different traits, because the only thing that the Y chromosome actually carries is how they produced testosterone, and so that a male can go through sexual differentiation. So if you see a male karyotype, you're going to look for the little tiny Y chromosome that doesn't match with any other chromosome, and then that would be that it's a male karyotype. So basically, when we're given karyotype information, we're going to give all of these scrambled up, and you have to match them up based on their size, banding pattern, and their centromere position. So this is another picture of something we can look at. So again, you would take all of the chromosomes from mom and all of the chromosomes from dad. They're paired up right here. And then this individual has all of them paired up now with their homologous chromosomes. Notice that they get smaller as they go down the karyotype, and then the last 2 chromosomes, which are the 23rd pair, this individual is XX, so it means that this cell is showing a female. So that's what we're going to be doing in building a karyotype. And I would say karyotyping is pretty important in this chapter, so it's kind of a big skill that you guys will have to do in this chapter and eventually be doing for our project. So that's why I'm getting you to practice today. So hopefully that makes sense. If not, please feel free to ask me questions, and I'll talk to you guys later. Bye.